Our today's topic is chromosomal theory of inheritance. Mendel, his findings were published in 1865 in a small natural history society in his native country, and his theory was not acceptable to his contemporary scientists because of so many reasons. Only in 1900, 3 different scientists rediscovered Mendel's principles. Now let us find out what were the reasons why the Mendel's principles were not accepted by the scientific community. Basically, Mendel was a teacher in a church school. He was a full-time preacher, part-time teacher. This is the first thing associated with not obtaining proper recognition by Mendel for his discovery of principles of inheritance. The first problem is that Mendel used statistical data to support his principles. This is the first problem. Second, Mendel used statistical data to support his principles. The second problem is he applied mathematical logic to explain the principles of inheritance, and this was unheard of to his contemporary scientists. The biologists of Mendel's age thought that mathematics and biology are different disciplines, and there is no way mathematical principles can be applied to biological phenomena. The third problem is Mendel explained his principles of inheritance based on discrete particles called factors, and there were so many variations in nature, and the discrete stable particles could not explain the so many variations observed in nature. And one more problem is Mendel could not explain or show where these factors are located. So factors, their location could not be explained by Mendel, and his brilliant theories were not supported by any kind of proof. This was the reason he could not get recognition during his lifetime. In 1900, 3 scientists independently rediscovered Mendel's principles. They were Hugo de Vries, Karl von Correns, and Sherlock. These 3 scientists independently rediscovered Mendel's principles, and they rediscovered Mendel's papers also. Hugo de Vries rediscovered Mendel's paper, and he published it in Flora in 1901. Karl Correns translated Mendel's papers. And formulated the principles of dominance and the law of independent assortment because Mendel used so many mathematical symbols, and it was difficult for his contemporary scientists, and Correns translated them and presented them in acceptable form. Now to the proof that the factors are located on chromosomes, this was supplied by 2 scientists, Sutton and Boveri. Sutton and Boveri, they supplied proof that the factors are located on the chromosomes, and that was called chromosomal theory of inheritance, and the chromosomes are considered as physical basis of inheritance. Chromosomes are considered as physical basis of inheritance. Now let us look into the similarities between the behavior of Mendel's factors and chromosomes. By the time of 1900, there was so much improvement in microscopy, and using microscopic studies, first mitotic behavior of chromosomes was studied. Later, meiotic behavior of chromosomes also studied, and Mendel's factors they appear in pairs, and cytological behavior basing on that chromosomes also appear in pairs. This is the first similarity. Mendel's factors appear in pairs, so also chromosomes during cell division also appear in pairs. This is the first similarity. And without blending, Mendel's factors segregate. Chromosomes also segregate. This is the second similarity. Chromosomes also segregate. This is the second similarity between factors and chromosomes. The third similarity, Mendel's factors assort independently. So also chromosomes also assort independently. So basing on the similarities, Sutton and Bowery proposed that the Mendel's factors are located on chromosomes. These are the similarities. Mendel's factors appear in pairs. Chromosomes also appear in pairs. Without blending, these factors segregate and enter gametes. Chromosomes also, without blending, segregate and enter gametes. And factors assort independently, so also chromosomes assort independently during gamete formation. These are the similarities. During sexual reproduction, what part is transferred through gametes? The male gamete. During sexual reproduction, there is fusion of gametes. The male and female gametes fuse. The male and female gametes fuse. The male gamete contributes what? Okay, let us think. Suppose this is the male gamete. This is the nucleus, and in most cases, this is the part that is transferred to the female gamete or egg. So, and what lies inside the nucleus? Chromosomes lie inside the nucleus. And the behavior of chromosomes from cytological point of view is like this. During G1 phase, the chromosomes during G1 phase will be having 2C quantity of DNA. During S phase, this becomes 4C, and during G2 phase, the same 4C DNA quantity will be there, and each chromosome from S phase onwards is having 2 chromatids. Each chromosome has 2 chromatids, and only one of the chromatid will be entering the gamete. Only half number of the chromosomes are entering the gamete. So, one genome will be present in the male gamete, another genome will be present in the male gamete and one genome in the female gamete. When these two fuse in the zygote, two genomes will be there, two genomes will be present in the zygote. During sexual reproduction, the zygote will develop into organism, the zygote will develop into organism. In the organism during gamete formation, meiotic division occur, producing gametes and each with half number of chromosomes or one genome only. One genome only. So, the chromosomes and factors appear in pairs, and during gamete formation they become half, and after fertilization the original number is restored. So, this is the cytological basis for presence of genes on chromosomes or factors present on chromosomes. Now, let us try to apply the chromosomal theory of inheritance to behavior of chromosomes during meiosis to dihybrid cross, where the parental cross is represented, representing round yellow crossed with wrinkled green seeded plants. This is the parental generation, and since it is homozygous for both characters, all its gametes contain capital R and capital Y only, and during gamete formation the recessive also will be contributing small r, small y in its gametes. The number of chromosomes become half. The alleles or factors appear in pairs, and during gamete formation only one of them will enter. That is what is called a law of purity of gametes. So, basing on law of purity of gametes, only one capital R and one capital Y, only one small r and one small y will be present in the gametes, and when they fuse we have F1 generation, where we have capital R, capital Y, capital R, small r, capital Y, small y. So, this is the F1 hybrid. This is the F1 hybrid. Let us consider this as a large sized chromosome with a gene locus, capital R here, small r here, and this is a small chromosome representing color with gene locus capital Y here and a small y here. So, since these are alleles, they are located at the same gene locus, and during gamete formation it will produce 4 different kinds of male gametes and 4 different kinds of female gametes. Now, the gametes will be like this. So, the Punnett square will appear like this, and the capital R, small r gametes will be capital R, capital Y, capital R, capital Y, and here we have longer chromosomes and shorter ones. It will be capital Y, and capital R, capital R. So, the other gamete will be capital R, small y, and this must be capital R, capital Y, small y. Here it will be small r, capital Y. So, the gametes must be, after fertilization will appear like this, and this will be capital Y, small y, and this will be capital R, small r. Third type of gamete combination will be capital R, small r, capital Y. The fourth type of gamete will be, so the gametes, when these eggs are fertilized by these male gametes, this is the condition, it will be capital R, small r, this is small r, small y. So, capital R, small r, and it is capital Y, small y. The F1 will be heterozygous with two different kinds of alleles in each case. So, this will be capital R, small r, this must be capital Y, small y. During gamete formation, there will be purity of gametes. What is meant by purity of gametes? Only one of the factors will be entering the gamete. So, in half of the gametes we are having capital R, in remaining half of the gametes we are having a small r. So, during gamete formation, we have four different kinds of gametes being produced. What is the reason for that? It is heterozygous for two pairs of characters. So, heterozygosity is 2 and possible gametes will be 2 to the power 2. Four types of gametes must be formed, and in half of the gametes there must be, basing on purity of gametes, law of purity of gametes, capital R must be present. In remaining half of the gametes, small r must be present. So, this is with regard to the first pair of alleles. With regard to second pair of alleles, half of the gametes must be having the capital Y and remaining half of the gametes must be having a small y, and they enter the gametes in random manner. So, there will be one independent, due to independent assortment, the capital Y will appear with capital R and also with small r. So, you have four kinds of gametes here, four kinds of male gametes. In similar manner, four kinds of female gametes also will be there. Four kinds of male gametes are here and four kinds of female gametes also will be here. So, the first kind of female gamete will be capital R, capital Y. The second will be capital R, small y. The third will be small r, capital Y. And the fourth one will be small r, small y. Now, so due to independent assortment, irrespective of parental combination, one gamete will enter the gamete, one chromosome will enter the gamete irrespective of parental combination. So, the first one will appear like this, and the zygote will be having the shape alleles as capital R, and this will be capital Y, capital Y. So, this is capital R, capital Y, capital Y. The second will be capital R, and this will be capital Y and small y, capital Y and small y. It will be its chromosome. This is the locus, gene locus for the shape of the seed. This is gene locus for color of the seed. So, this is capital R, capital Y, small y. With regard to third kind of zygote, that is produced randomly, there is capital R, small r. So, there is capital R, small r. So, on the homologous chromosome we have small r. Similarly, it will be capital Y and small y at the other gene location. So, this is capital R, capital Y, small y. Capital Y, capital Y. It is, because of this condition, it is homozygous. So, capital Y, capital Y. With regard to third kind of zygote, we have two alleles for shape and two alleles for color. So, this must be small y, capital Y and small y, and this must be capital R and small r. So, this will be capital R, capital Y, small y. Similarly, it will be capital Y, small y and capital R, capital R. So, this is capital R, capital Y, small y. Similarly, this will be capital R, capital R and small y here. So, this must be capital R, the other will be small y. With regard to this will be capital R, small r and capital Y, small y, and the chromosomes will be like this. This will be capital R, this will be small r and capital Y, and the other will be small y. With regard to the genetic combination of this zygote, it will be capital R, small y, small y. So, the gene loci will appear like this. This will be capital R and this will be small r, and for color it is small y and small y only. The remaining also will be similar here. The remaining gametes also will be similar here, and it will be capital R, capital Y. So, the gametes must be, this is capital R, and this must be capital Y. With regard to the combination here, it will be capital R, capital Y, small y. So, this must be capital R dominant one, and this is small r, and in this case this is capital Y, this will be small y. Similarly, it will be small r, capital Y. So, here both will be recessive for shape and both will be dominant for color. With regard to the other one, we have small r and capital Y, small y. With regard to shape, this will be recessive. With regard to color, one will be dominant, other will be recessive. Now, the last one will be like this. This is capital R, capital Y, small y. So, the chromosomes will be like this. This is dominant capital R, this is recessive small r, and this is dominant capital Y, this is recessive small y. Then here we have capital R, and it is small y, and the gene loci will be capital R, and this will be small y and small y. With regard to the other one, it will be small r, capital Y, small y. So, the shape genes will be recessive, the color genes will be one dominant, other recessive, and this will be double recessive, small r, small y, and with regard to this it is small r, and with regard to color it will be small y. So, during gamete formation, chromosomes show law of segregation or purity of gametes. Out of the two in the parent, only one will be entering. Out of two alleles, only one allele will be entering the gamete. This is, and during, so what are the parents here? Capital R, capital R, capital Y, crossed with small r, small r, small y. So during gamete formation, R will enter a gamete with capital Y and also a gamete with a small y. Similarly, capital Y will enter a gamete having capital R or small r, of course, both in equal number. So 50% will be joining the capital R, remaining 50 will be joining the small r. This is what is law of independent assortment. Sutton and Bowery proposed chromosomal theory of inheritance. The actual proof was provided by Thomas Hunt Morgan. T.H. Morgan proposed and supplied the proof for chromosomal theory of inheritance. He studied the fruit fly, common fruit fly, Drosophila, melanogaster. There were certain advantages associated with choosing this particular organism. Fruit fly can be grown on synthetic medium. Fruit fly can be easily grown on synthetic nutrient medium. It can be, this is the first advantage, it has well defined characters, so many variations are there, and male and female are different. During each mating, large number of progeny produced. During each mating, large number of progeny produced, and they completed their life cycle in just 2 weeks. So, within a short span of time, many generations of fruit flies can be studied conveniently, and their characters can be easily observable under light or simple microscope. So, characters can be studied using a simple microscope. So, Thomas Hunt Morgan and his associates started working with fruit fly Drosophila melanogaster. This can be easily grown on synthetic medium, and it has well defined observable variations or characters. Male and female are different. Male is larger and female is shorter, and during each mating they produced a large number of progeny, and their life cycle is very brief. It is completed in 2 weeks, and the characters of the organism fruit fly can be studied conveniently under a simple microscope. So, using the fruit fly, T.H. Morgan proved that the genes are located on chromosomes. Genes are located on chromosomes. This is the proof supplied by Thomas Hunt Morgan. Select the correct statement. Now, let us look at the question given in 2010 AIPMT examination. Select the correct statement. Tightly linked genes on the same chromosome show higher recombinations. Genes far apart on the same chromosome show higher recombinations. Genes loosely linked on the same chromosome show similar recombinations. Tightly linked genes on the same chromosome show very few variations. This is the fourth one is the correct option here, our answer being 4. Now let us conclude what we discussed today. Now let us try to conclude what we discussed today. The chromosomal theory of inheritance. The chromosomal theory of inheritance was proposed by Sutton and Boveri. Chromosomal theory of inheritance was proposed by Sutton and Boveri, and the actual proof was supplied by Thomas Hunt Morgan. Chromosomes as well as Mendelian factors both show segregation, both appear in pairs, and both behave independent of others in the sense Mendel's factors are genes and chromosomes assort independently. During gamete formation, chromosomes of both parents randomly enter gametes. Now let us consider some objective questions given in different examinations basing on monohybrid cross, dihybrid cross, mutations, linkage, crossing over, and also incomplete dominance and codominance. వివిధ పోటీ పరీక్షల్లో ఇచ్చిన ప్రశ్నల్ని ఏకసంకర సంకరణం, ద్విసంకర సంకరణం, ఉత్పరివర్తనాలు, సహలగ్నత, వైవిధ్యాలు వంటి పాఠ్యాంశాల నుంచి పరిశీలిద్దాం. This is given in 2007 examination basing on R and Y genes in maize lie very close to each other. When RRYY is crossed with RRYY, F2 generation will show segregation into in 9:3:3:1, segregation in 3:1 ratio, higher number of parental types, higher number of recombinant types. మొక్కజొన్న మొక్కలో RY జన్యువులు సమీపంగా ఒకే క్రోమోజోమ్ మీద సమీపంగా ఉన్నాయి. క్యాపిటల్ RRYY జనక మొక్కను స్మాల్ RRYY మొక్కతో సంకరణం చేయగా ద్వితీయ సంతతి తరం F2 జనరేషన్లో వచ్చే నిష్పత్తి ఏ విధంగా ఉంటుంది? ఇవి పృథక్కరణ 9:3:3:1లో చెందుతాయి. పృథక్కరణ 3:1గా చెందుతాయి. ఎక్కువ జనకతరం లక్షణపు మొక్కలు ఉంటాయి. ఎక్కువ సంఖ్యలో పునఃసంయోజనాలు చూపించే మొక్కలు ఉంటాయి. To obtain proper answer for this, This represents linkage here, and the R and Y genes are linked, and once R and Y genes are linked, higher parental combinations will be there, fewer recombinations. This will be fewer, that will be always higher. ఇవి రెండు సహలగ్నత చూపడం వలన జనకతరం లక్షణాలు అధికంగా కలిగినటువంటి సంతతి తరం వస్తుంది. అదే రకంగా పునఃసంయోజనాల శాతము లేదా సంఖ్య పరిమితంగా ఉంటుంది. సో సహలగ్నత మీద ఇవ్వబడిన ఈ ప్రశ్నకి హయ్యర్ నెంబర్ ఆఫ్ పేరెంటల్ టైప్స్ ఇది కరెక్ట్ ఆన్సర్. సహలగ్నత మీద అడగటం వలన ఎక్కువ జనకతరం లక్షణాలు కలిగినది అనే జవాబును తీసుకోవాలి. నెక్స్ట్, దిస్ ఈజ్ గివెన్ ఇన్ 2009 AIPMT examination. Point mutation is loss of gene, change in the base of gene, addition of a gene, deletion of a segment of gene. Here, పాయింట్ మ్యుటేషన్ అనగా పాయింట్ ఉత్పరివర్తనం అంటే జన్యువు కోల్పోవడం లేదా నత్రజని క్షార జతలో ఒకటి మారడం, ఒక క్షారం మారడం, జన్యువులోని ఒక క్షారం మారడం. అదే రకంగా అదనంగా ఎడిషన్ ఒక జన్యువును చేర్చటం లేదా తొలగించటం, దీన్ని ఎడిషన్. అండ్ దిస్ ఈజ్ ఇన్సర్షన్, డిలీషన్. ఇది కూడా లాస్ అవుతుంది. కరెక్ట్ ఆన్సర్ మస్ట్ బి చేంజ్ ఇన్ ది బేస్ ఆఫ్ ఎ జీన్. చేంజ్ ఇన్ ది బేస్ ఆఫ్ ఎ జీన్ ఈజ్ ది సూటబుల్ ఆన్సర్ in this case. Now, this is given in 2010 AIPMT examination. This is our question. Which are the following cannot be explained on the basis of Mendel's law of dominance? Now the options are, out of one pair of factors, one is dominant and other is recessive. Out of one pair of factors, one is dominant and other is recessive. Because it is law of dominance, this exactly says in hybrid condition, one is expressed and other is not expressed. Now let us go for the second option, and here we have alleles do not show. Alleles do not show any blending and both characters are recovered. Both characters are recovered. As such in F2. As such in F2. Now let us go for the third and fourth options. Third and fourth options here. The third, factors occur in pairs. factors occur in pairs. Let us take up the fourth option. Discrete unit controlling a particular character is called factor. బహిర్గతత్వ నియమం ద్వారా ఈ క్రింది వారిలో దేనిని వివరించలేము? వన్ ఫ్యాక్టర్ ఈజ్ డామినెంట్, వన్ ఈజ్ రెసెసివ్. ఒకటి బహిర్గతత్వాన్ని, ఇంకొకటి అంతర్గతంగాను ఉండిపోతుంది. ఇది మనం లా ఆఫ్ డామినెన్స్ నుంచి చెప్పచ్చు. ఎలీల్స్ డు నాట్ షో ఎనీ బ్లెండింగ్ అండ్ బోత్ క్యారెక్టర్స్ ఆర్ రికవర్డ్ యాజ్ సచ్ ఇన్ F2. The second option is based on what is called law of purity of gametes or law of segregation. Law of purity of gametes or law of segregation. So it has nothing to do with the law of dominance. ఇది పృథక్కరణ సిద్ధాంతం ఆధారంగా చేయబడినటువంటిది. సో సెకండ్ ఈజ్ ది కరెక్ట్ ఆప్షన్. పృథక్కరణ సిద్ధాంతాన్ని అనుసరించి చేయబడిన రెండవ జవాబు ప్రత్యామ్నాయం సరి అయినది. నౌ లెట్ us take up few more questions from different aspects here. In antirrhinum, 2 plants with pink flowers were hybridized. The F1 plants produced red, pink and white flowers in the ratio 1:2:1. What would be the genotype of the parent? Parent plants. RR is red and RR is white. This is given in AIPMT 2010 examination. యాంటీరైనం లేదా డాగ్ ఫ్లవర్ మొక్కలో 2 పింక్ వర్ణపు పుష్పాలను ఏర్పరిచే మొక్కలను సంకరణం చేయగా F1 తరంలో ఎరుపు, పింక్ మరియు తెలుపు పుష్పాలు కలిగినటువంటి మొక్కలు 1:2:1 నిష్పత్తిలో ఏర్పడినాయి. అప్పుడు ఆ మొక్క యొక్క పింక్ వర్ణం కలిగిన జనకతరం మొక్కల యొక్క జన్యు రూపము ఏమై ఉండవచ్చు? ఇక్కడ సూచనగా క్యాపిటల్ ఆర్ ఎరుపు వర్ణాన్ని, స్మాల్ ఆర్ తెలుపు వర్ణపు పుష్పాలని ఏర్పరుస్తుంది. దిస్ క్వశ్చన్ ఈజ్ బేస్డ్ ఆన్ ఇన్కంప్లీట్ డామినెన్స్ అండ్ సిన్స్ ఆర్ఆర్ విల్ బి వైట్, దిస్ ఆప్షన్ ఈజ్ నాట్ సూటబుల్. స్మాల్ ఆర్ తెలుపు వర్ణాన్ని ఇస్తుంది కాబట్టి ఇది జవాబు కాదు. సెకండ్ ఆప్షన్ క్యాపిటల్ ఆర్ స్మాల్ ఆర్ అండ్ దిస్ ఈజ్ ఇన్కంప్లీట్ డామినెన్స్. అసంపూర్ణ బహిర్గతత్వంలో మనకి కొత్త లక్షణం, జనకతరాల్లో లేని కొత్త లక్షణం వస్తుంది. పింక్ వర్ణం కలిగిన పుష్పాలు దానికి ఉదాహరణ కాబట్టి క్యాపిటల్ ఆర్ స్మాల్ ఆర్ సరైన జవాబు. నెక్స్ట్ థర్డ్ ఆప్షన్ capital R, this will give us white, this will give us red. So 1 and 3 out of place. Next, fourth option RR. This also will produce white flowers only. అంటే మనకి అసంపూర్ణ బహిర్గతత్వం వ్యక్తం చేయబడుతుంది. దీని నుంచి తెలుపు వర్ణ పుష్పాలు, దీని నుంచి ఎరుపు వర్ణ పుష్పాలు, దీని నుంచి కూడా తెలుపు వర్ణ పుష్పాలు ఏర్పడతాయి. ఇది విషమ యుగ్మజ స్థితిలో అసంపూర్ణ బహిర్గతత్వం జరుగుతుంది కాబట్టి మన జవాబు క్యాపిటల్ ఆర్ స్మాల్ ఆర్ ఉన్న రెండవ జవాబు సరైనది. నెక్స్ట్ క్వశ్చన్, దిస్ ఈజ్ గివెన్ ఇన్ 2011 AIPMT examination 2011. What is the question? All the 7 children of a couple are male. What is the probability? What is the probability that the 8th child will also be a male? This is basing on probability of progeny. Here, what are the two probable progeny for any couple? Either male or female. So total outcomes are 2 and the result will be 1 out of 2. So the probability will be male has a choice of 50% choice, female also will be having 50% choice. So this must be the proper answer, and what is given here is 1 by 2. This is our answer. The second is 1 by 4 and the third is 1 by 8, the fourth being 1 by 16. Total events are 2, either boy or a girl, and the individual event will be 1. So the chance of getting a male child will be 50%, the chance of getting a female child will be 50%. ఒక జంటకు ఉన్న మొదటి 7 సంతానము బాలురైన 8వ సంతానము బాలుడు అవడానికి ఉండే సంభావ్యత ఎంత? ఇది ప్రాబబిలిటీ సిద్ధాంతం ఆధారంగా ఇవ్వబడినటువంటి ప్రశ్న. మెండల్ టీటీ గ్యామెట్స్ విల్ ప్రొడ్యూస్ 50% T and remaining 50% small t. So here this is boy and this is girl, or this is girl and this is boy, the choice will be 50%. So సంభావ్యత నియమం ప్రకారం 50% బాలుడు జన్మించే అవకాశం, మిగిలిన 50% బాలిక జన్మించే అవకాశం ఉంది. సో 50% is our correct answer. Next, this is given in AIPMT 2012. This is the question. A test cross is performed. A test cross is performed to know genotypes of F2 dominants, linkage between two traits, number of alleles in a gene success of intervarietal and interspecific crosses. Now, this is given in 2012 examination. పరీక్షా సంకరణం దేన్ని తెలుసుకోవటానికి చేస్తారు? పరీక్షా సంకరణం దేన్ని తెలుసుకోవటానికి చేస్తారంటే, మనం ఒక బహిర్గత లక్షణం చూపించే మొక్క యొక్క జన్యురూప స్థితి తెలుసుకోవడానికి దీన్ని ఉపయోగిస్తారు. సో బికాజ్ డ్యూ టు డామినెన్స్, all dominant plants, homozygous dominant and heterozygous dominant will be similar. To know whether they are homozygous or heterozygous, a test cross is performed. So first option is the best option. జన్యు స్థితిని తెలుసుకోవడానికి పరీక్షా సంకరణాన్ని నిర్వర్తిస్తారు. ఇది మొదటి జవాబులో మనకి కనబడుతోంది. Now, one more question given in 2012 examination. This is the question, and let us check the question. AIPMT 2012. Both alleles express in heterozygote when they are first option recessive, lethal, semi-dominant, fourth option codominant. Now, రెండు యుగ్మ వికల్పాలు విషమ యుగ్మజ స్థితిలో వ్యక్తం చేయబడేది అంతర్గత లక్షణం, హానికరం, సెమీ డామినెంట్, అసంపూర్ణ బహిర్గతత్వం, సహ బహిర్గతత్వం. దిస్ ఈజ్ ది బెస్ట్ ఆప్షన్. A recessive is not expressed in heterozygote. Lethal gene will cause death of the progeny, so there is no question of expression. A semi-dominant gene causes incomplete dominance. It causes incomplete dominance, whereas codominance, both characters find expression in case of codominance. So that is the best and correct option in the for the given answer. Now, one more question given in AIPMT 2012 examination. Among seven pairs of traits studied by Mendel, among seven pairs of traits studied by Mendel, the number of traits related to flower, pod and seed. Now, these are the options here. The first one, 2, 2 and 2. The second one, 2, 2 and 1. The third one, 1, 2 and 2. The fourth one, 1, 1 and 2. Let us consider how many characters among 7 characters are related to flower. So the flower characters are location, axillary or terminal, color, that is violet or white. So 2. Next, this is pod. Regarding pod, we have shape, constricted or inflated, color, it is green or yellow. So second is also 2, either this or this. This and this not possible. Next we have seed. Seed characters, shape, round versus wrinkled, and color, yellow versus green. So 2 here, 2 here and 2 here. First option is the best option. Now, let us consider మెండల్ పరిశీలించిన 7 లక్షణాలలో పుష్పము, ఫలము, విత్తనాలకు సంబంధించిన లక్షణాలు ఎన్ని అంటే పుష్ప లక్షణాలు గ్రీవస్థము లేదా శిఖరస్థము అనే ఒకటి, వర్ణము ఊదా వర్ణము లేదా తెలుపు అంటే 2 లక్షణాలు, ఫలముకు సంబంధించి కన్స్ట్రిక్టెడ్ ఆర్ ఇన్ఫ్లేటెడ్ అండ్ ఆకుపచ్చ అంటే వర్ణానికి సంబంధించి ఆకృతికి సంబంధించి 2, విత్తనానికి సంబంధించి ఆకృతి వర్ణము 2. మొదటి జవాబు సరిపోతుంది.